Okay, but it's hard to imagine this little person in all your cells doing that. And cells don't really have brains, so what are they thinking about? And so there's actually elegant, very simple systems that have evolved that act as checkpoints that can basically sense whether the environment is right, everything is set, and then you can go to the next step. And so one of these is something called the anaphase promoting complex, which literally does this. It promotes anaphase. It promotes the transition from metaphase to anaphase. So it involves what's called a spindle checkpoint. So when all the chromosomes are lined up and they have the microtubules attached properly, what's happening is the microtubules are pulling a little bit, and now tension is occurring in those microtubules. And when there's tension on all of the microtubules, okay, then that activates this protein. It's actually a complex of proteins called the APC. And then that APC is activated. It degrades a protein called securin. And securin is actually holding on to separase. Okay? So when securin gets degraded, separase is released and active. And then when separase is active, it cleaves cohesin. Okay? And so now the chromosomes can come apart. So what it means is that the chromosomes are stuck together until essentially the cell knows that all the microtubules are attached properly. And it knows that because there's tension now in every one of those microtubules. Right? And so then the APC is activated, and that can go to anaphase. And so that's sort of a simple idea of a checkpoint. We're simplifying it a lot here. But that's the basic idea. And there are lots of these kinds of checkpoints. So for example, you don't want to start actually condensing chromosomes while you're still replicating your DNA. And so there's a checkpoint that you don't actually condensate until S phase is really done. Okay? Because if you did, you could then start breaking the DNA that was or wasn't replicated yet. Okay, so, and this helps to avoid non-destruction during mitosis. Oops, I should say, but what about meiosis? Okay, so there's an interesting problem at meiosis, which is how do you create this tension? Because remember that cohesin is holding together the sister chromatids, but at meiosis 1, you have to line up these non-sister chromatids, right? And so they're not really stuck together, right, by cohesin. So how are you going to get the microtubules to attach and then get any tension? Okay? And that actually is one of the functions of the crossovers that occur. So remember, we described these crossovers to you as how you get recombination. And so recombination is useful because it allows you to independently segregate genes that are on the same chromosome, right? But it also serves another important function, which is then that forms the link between those homologous chromosomes so that when they're pulled on, they're actually stuck together. And they're stuck together because of those chiasmata, right, where the chromosomes have crossed over. And so then when you pull on it, they're stuck together. That's why, in fact, there's always at least one recombination event on every arm of every chromosome. Okay? That's why you don't get non-recombinant chromosomes. Because it has to happen. So what in fact is going to happen is the cell is going to wait until there's at least one crossover on every arm of every chromosome. Because only then will there actually be tension on all the microtubules. So once at least one crossover is initiated, now there's tension, and now you can start pulling them apart. Okay? And then they're going to resolve when they get pulled apart. So, and that allows that to happen. You don't have to worry about that at meiosis 2 because meiosis 2 you again have cohesin holding the sister chromatids together. Okay? So that also then gives you another interesting problem because what about X and Y? So for all autosomes this is fine because you can understand how you get crossovers between them. But X and Y aren't homologous chromosomes. They don't actually have similar sequences on them for the most part, right? But that turns out to be, people discovered, well, that's not totally true. So it turns out that there is a region of homology on X and Y, between X and Y, but it's at the tips of X and Y. And it's in regions where there are no genes. Okay? So you actually do get crossing over between X and Y, but there's no genes in that region. And so you don't ever see that from genetics if there's anything going on. But molecularly that happens, and that's required in order to get tension of the XY pair. Okay? Okay, so that's how you manage to do that. And that only is a problem with meiosis. Mitosis is not a problem because X and S are together and Y and Y are together in a male. Right? I don't actually know whether that's true. I don't know if it's true. It's also possible that meiosis actually has a stronger checkpoint so that it would avoid that problem. But it may not because if the result of that is a mistake, there's other mechanisms to kill the cell. I should point out, we didn't put this on a slide, but so what happens if a cell gets caught and it just can't pass a checkpoint? Okay, so like the chromosome is lost and it doesn't have a pair. It's never actually going to get through that checkpoint. And so cells that get stuck at checkpoints initiate something called apoptosis. Have any of you heard of that? They actually initiate programmed cell death and the cell will kill itself. Okay, and this is actually a good thing because if the cell did manage to divide, it would have a chromosome defect in it. So the cells that get stuck at checkpoints will commit suicide and get rid of themselves, which is to the benefit of the whole organism to do that. Okay. Okay, so there's another problem though with the sex chromosomes, and that's an issue of dosage. So in humans, right, you have XX versus XY. And that means that you have different doses of chromosomes on the X chromosome. Okay, for most genes that would be okay. But there's a few genes, again, that we call haploinsufficient. You really need 2 copies, okay? Or you need to control the dose, sorry. The more accurate things, you need to control the dose. And so in mammals, what you do is you actually make it so that there's only one active X per cell. Okay, so that gets the dose to be the same between females and males. Okay? And so the way you do that is through a process called random inactivation. So in females, in individual cells during development, what you'll do is you'll inactivate one of the 2 X chromosomes. Okay? So in a woman, right, they're actually mosaics of which chromosome is active. And so that can lead to some interesting patterns. If the X chromosome is heterozygous for some locus, it means that there'll be one allele in some cells and another allele in the other cells that are active. And so that's actually how you get, for example, patterns on cats, coat color patterns for some of them. Because if you imagine that you have a gene that controls the difference between orange and black fur, but it's on the X chromosome, what happens during embryogenesis is that we randomly inactivate one or the other. And then that pattern is continued. So in this cell and all the progeny cell, only the black fur allele is active. And in this cell and all its progeny, only the orange fur. And this happens when there's a few thousand cells, okay, total in the embryo. And so then if you look at the surface of the cat, it's a patchwork of orange and black fur. But it also leads to something interesting, which is that if you have twin cats that are genetically identical for a gene like this, they won't have the same pattern of coat color because it's caused by random inactivation. So there's no set pattern to that inactivation. So you can, in fact, there's a famous example where the first cloned kitten, right, was a pure clone from another cat. It was a calico cat, but they look completely different from one another, even though they're genetically identical. And that's because it had a gene that was inactivated on the X, different alleles. Okay. You should think about how this would also affect epistasis. So if the gene is interacting with other genes on the autosomes, you can get very complicated patterns. It occurs when the embryo is still quite young, so there's a few thousand cells. Okay, and then it occurs. And at that point, the primordia of the skin is maybe 10 or 20 cells, and so you get this random patchwork. Okay. It doesn't happen in males. Males only have one X. They don't inactivate the one X, so the cell would die. Okay. Okay. So, oops, sorry. All right. So one of the things though that we can do then is because we can use mistakes like this to actually follow where cells are going. So we said, for example, that inactivation in females occurred when there was 1,000 cells, or maybe about 10 making the coat, the ectoderm. And so then the cells that are similar colors that are together, that's because they came from a single cell in the embryo, and those progeny of those cells stayed close together. And that's also what's going on in these wings. The reason that they're in these large patches is because it's a rare event, and then the progeny of those cells stay together. And in fact, one of the things we do in my lab is study how we can tell how the wing is being made by looking at the pattern of those clones. And so if you look at human X inactivation, the same thing actually happens. So there's a few very rare conditions in females where, again, there's mosaicism of pattern on the skin because there's an allele controlling some aspect of skin color or the presence of sweat glands, and then you get these amazing patterns. on these women. And what these patterns are showing you in this case is how cells migrate when the skin is being formed. And so you get what are called Blaschko lines. There was a German scientist long ago who discovered this. And this is an example of a woman that actually has one of these conditions. And the reason that you get these patterns is because during embryonic development, that's the way the cells are migrating and dividing and spreading out. And so we can use this as what we call a lineage marking technique to tell where cells are going. Okay? And then finally in flies, this doesn't happen. And the reason it doesn't happen is that dosage compensation works in a completely different way. So in flies, what you do is that when you're XY, you actually upregulate transcription off the single X to match the level of transcription that you would get from two X chromosomes. And we don't have time to go into that mechanism. Okay? But just to illustrate that even though they're XY, they still do things in a different way. Okay, so now in the very last bit of the class, we'll just talk about one last issue, which is another reason to have checkpoints, right? And that's these are important to avoid cancer. And so cancer is not a single disease, right? Rather, it's a broad way we have of defining anything where there's an uncontrolled growth of abnormal cells in the body. Okay, and the severity then of a cancer depends on how abnormal the cells are and how fast they're growing and whether they are spreading right outside of where they started. Okay, and probably most of you are familiar with these kinds of concepts for cancer. So how do you end up with a cancerous cell? So for example, how do you end up bypassing growth control? So again, there's a lot of checkpoints that control when cells enter it and progress through mitosis. Okay, and if you bypass those checkpoints, you start getting chromosome abnormalities. You start dividing too fast, and for example, if you don't make sure you're at that metaphase to anaphase transition, then you start pulling chromosomes apart. And in fact, that's one of the hallmarks of cancer cells is they have lots of messed up chromosomes in them. Okay, so one of the ways that this sort of thing can be activated is there's a protein called PDGF or platelet-derived growth factor, and there's a receptor for that. Okay, and what will happen sometimes is that mutation will occur that will cause cells to overexpress PDGF, and PDGF is one of the signaling pathways that tells a cell it's okay to go ahead and start dividing. So normally what happens is cells differentiate, they stop responding to PDGF, they stop making PDGF, and then they'll stop growing, they'll stop dividing. Right? You need that to occur. But if you have a mutation that causes this to be overexpressed, now you've got a problem. The cells are just going to keep dividing, and so they get by the checkpoint because they're overexpressing this. So one way you get cancer is, for example, a mistake in this gene that causes it to be overactive. Like the receptor becomes activated, it thinks it's bound all the time, and it starts making too much signal, gets by all the cell cycle checkpoints, and then you get uncontrolled division. Okay? Sometimes cancers are caused by viruses, and that seems like a very odd concept, right? Why would a virus cause cancer? But there are a couple cases where that's very clearly true, that cancers can be caused by a virus. And a well-known example is HPV virus in women. And in fact, it's led now recently to an immunization program to prevent cervical cancer, because this is a major cause of cervical cancer, this particular virus. But how does a virus cause cancer? What's the virus going to do? And so it turns out that what happens is that viruses will occasionally essentially steal a gene from their host. It's an accident. When they're packaging up the viral genome, they accidentally package up an existing gene from a chromosome. Okay? And usually that's of no use to the virus. In fact, it's to the detriment of the virus. But every now and then it can take up a gene and end up with and either then it'll mutate in the virus or it take up a mutant copy of the gene. And there are a few genes then that are what we call oncogenes. That they're genes that when they're in a mutant form, they can cause cancer. And so there's a number of them. There's a couple dozen of them. But here's just one example, a very famous one called RAS. And so RAS is a protein that's involved in many different instances where growth factors or other signaling molecules are signaling to a cell. And then RAS is activated and it ends up acting in a pathway and then causes a lot of downstream targets to turn on and off. Okay? They can control cell division and so on. So normally RAS is very tightly regulated, so that only at certain times does it get activated. But occasionally, right by chance, you can get a mutation in RAS that causes it to be hyperactive. Okay? And now the cell goes out of control because the RAS is continuously signaling, and then this pathway is continuously on, and then the cells start dividing out of control, and they also stop doing their normal functions. Okay? Whatever they were supposed to do, like liver cells or something like that, they just stop doing any of that. So what happens is that you can get spontaneous mutants in RAS. So sometimes that's what causes a spontaneous cancer. Okay? It can be a spontaneous mutation or one that's induced by chemical damage. But in the case of viruses, sometimes the viruses can pick up a RAS gene and then have a mutant copy or mutate in the virus. Right? And now this is actually to the benefit of the virus, because if the virus has what's called activated RAS, this mutant copy of RAS, and it infects a cell, then that cell actually starts to divide really quickly. And then that means that there's a lot of virus-infected cells, so that helps the virus spread. Okay? But of course, the big downside to you is that gives you a cancerous cell, which starts to spread. Okay? And so there are quite a number of genes like this. People get confused by this because they think, well, they call cancer genes, right? But you have copies of RAS, and you absolutely need RAS to live. Okay? So it's not a cancer gene. It serves a very specific normal function. But what you have is a mutant allele of RAS that's not doing what it's supposed to do, and that's what's an oncogene. That's the cancer gene. Okay? So it's always a problem in the press when you read about, if they clone some new cancer gene or some gene that causes this disease. It's really an allele that causes the disease. The gene is probably there for some good function that it's got to serve, and it's a mutant copy that's doing this. Okay? So that's going to be it for lecture now, and I just thought I'd summarize very quickly for you what's going to be on the exam, so you were prepared for that. Okay, so it's 35 questions. So I tried my best to try to make sure it wasn't too long. So each question is worth somewhere between 2 and 5 points, and the question will say how many points it's worth. And of those, I think that 23 are a relatively quick answer. They're things that aren't really problems, and you should be able to come up with the answer relatively quickly. And then the remaining are more complicated, but there's not a lot of complex genetic problems. Okay, most of them are sort of experimental scenarios that are going to require you to combine information from different things that you've learned in lecture together, and then come up with an answer. Again, it's all multiple choice then. Okay, all right, thanks a lot.